everyone, welcome back to another histology walkthrough with Professor Howard. It has come to my attention that for some reason the histology video I made for the esophagus has winked out of existence and I can't find it, so I'm going to make another one. So, what we'll do in this video is describe the general histology of the esophagus and how you can recognize it as being the esophagus and make sure you can distinguish it from other structures, because that's the goal, right? You want to be able to look at a picture like this and say, ah, that checks all of my boxes for esophagus, that must be what it is. So as I always recommend, we want to start zoomed out. I know that there's temptation to immediately zoom as far in as you possibly can because students kind of associate zoomedness with detail. But you have to learn the lay of the land first and make general observations so that what you see when you zoom in has meaning or context. If you zoom in too soon, you'll get lost in the weeds, and that's not what we want here. So let's have a look at what we see here. So I'm going to just kind of summarize my general observations over to the right. So it's definitely a hollow organ, and it's got kind of a collapsed lumen. Another word for this is stellate lumen, stellate meaning star-like or star-shaped. As you can see, it's not really star-shaped, but it's just reflective of the fact that it kind of has collapsed into a structure with points on it. So it's hollow, it's got a collapsed lumen. I can see what appear to be glands. I'm going to have to confirm that by moving further in, but nice initial observation. And then I can also see what appears to be some discrete layers. If it's a hollow organ, that means that it will be lined with epithelium of some kind. And then I also think that I see some dense irregular connective tissue and what looks like smooth muscle out here. And one final observation I can make at this magnification is that the epithelium appears to be stratified. Okay, so that's a pretty good start. So if I'm, thinking about what this could be right off the bat before I zoom in, as a student, that's a skill that you need because time is of the essence when you're taking a practical, and so quickly and efficiently narrowing down what you could be looking at and eliminating other possibilities is always a go-to. So if I'm preparing to answer a question about this, I would say, well, I don't see any hyaline cartilage present, so it's obviously not the trachea and it's not a bronchus. I don't see any lung tissue. I do see glands, which suggest that whatever moving through this tube has to be lubricated in its passage. And also, since I see stratified epithelium, that means it's not a blood vessel. So that's me kind of modeling for you kind of how you can use what you observe to eliminate possibilities and approach tissue identification and organ identification from a logical series of steps. Instead of what I call the stare and cry method, which I think I've mentioned in previous videos, which is where you try to learn histology by staring at unfamiliar images until hopefully you recognize them, and then crying when you arrive at the practical and you can't. So that's what we want to avoid. Okay, so now that I've made my initial observation, let's go zoom in. And I'm going to start by having a look at the lining, and let's actually zoom in a little bit more. Okay. So what I'm seeing about this lining is it's quite thick, so it is indeed stratified, and if I have a look at the cells at the apical surface, I can see that they are thin and flat, which means that this is stratified squamous epithelium. Okay, that's a good observation. So now let's have a look at the other structures I observed. So what we're going to do now is just kind of cruise around the slide and see what there is to see. This behavior, starting zoomed out, making observations, and then cruising around and seeing what there is to see, is perhaps the number one mistake that I see students making, and what I mean by that is not doing that. So watching these videos is awesome. It's of course important to get an initial handle on what you can expect from a tissue or an organ, but if you're not practicing histology by going to Histology Guide and navigating around yourself and seeing what observations you can make and what structures you recognize, you're really doing yourself a disservice as far as preparation for the practicals. So I strongly recommend doing that. Okay, so I see that my epithelial lining ends about here, and then I see two different tissues. So there's this distinct band, and I'm going to highlight it in a different color. Let's highlight it in orange. Why not? So I'm going to just kind of highlight what I'm seeing here. Let's use a bigger dot. So this band of tissue here is distinct and different from what's surrounding it. So, looking at other stuff in the field of view here, I can see this is definitely dense, irregular connective tissue. I can see an artery and another artery over here. I can see a vein, and I see the nuclei of fibroblasts. And then I also have this little area of connective tissue between the epithelium and this orange band. So the orange band is smooth muscle. And so what that tells me is that I'm looking at the Muscularis mucosae. So I want to pause here and make an important observation about the way students show me that they think about histology. Smooth muscle is the tissue type. And you can find smooth muscle all over the place. This particular arrangement of smooth muscle, where it's a narrow band of smooth muscle between the epithelium and the submucosa, which is what this layer is, tells me that the structure that I'm looking at that's made out of smooth muscle must be the Muscularis mucosae. So this is an interpretation problem that students have, where when I ask for something like identify the specific structure highlighted in orange, what I'm asking you to tell me is what its specific name is in the context of this organ, not what tissue type it is. I am acknowledging that it's smooth muscle, and that's because the Muscularis mucosae of any organ is made out of smooth muscle. Noticing that it's smooth muscle and noticing its position in the tissue is how you arrive at the conclusion that it's the Muscularis mucosae. So both of those pieces of information are important, but they're at different levels of specificity. All right, so I've located my Muscularis mucosae. I'm going to delete the highlighting for a moment. just so that you can have a little peek at what it looks like when it's not highlighted. And so this is one of the examples of a time where the more examples of a tissue you look at, the easier it will be for you to recognize it, because here we have kind of a mixture of cross-sectional and longitudinally sectioned smooth muscle cells. So it's not entirely longitudinal, which is what students tend to look for, those long spindle-shaped cells, and it's not always going to be. So what I'm noticing is it's a little bit more purple than the very, very pinkish red, dense, irregular connective tissue, and I can see that the groups of muscle cells are separated from each other, but overall it definitely gives the appearance of being smooth muscle. So again, practice, practice, practice. The more smooth muscle examples you see, the better you will become at identifying it. Okay, so I'm going to zoom out a little bit. So I can see that the submucosa is here. And remember, the submucosa is under the mucosa. So the mucosa consists of mucosa is a way to refer to a mucous membrane. So the mucosa consists of epithelium, an underlying lamina propria, which is a narrow band of connective tissue, typically loose connective tissue, that is between the epithelium and the muscularis mucosae. So the lamina propria is right here, I'm bracketing it, and then the muscularis mucosae. So mucosa, I've got my submucosa here. The submucosa of hollow organs tends to be where you find the majority of blood vessels and glands, although I don't see any of the glands in this field of view. We'll have to go elsewhere for that. And then out here I see a mixture of different views of smooth muscle. And the smooth muscle is the smooth muscle of the layer called the Muscularis externa. So this mucosa, submucosa, muscularis externa naming scheme is a way to refer to the layers or tunics of hollow organs, primarily those of the GI tract. So even if I didn't have all these helpful labels here telling me what I was looking at, I would still come to the conclusion that I was in the GI tract, because if I observe carefully, I can see those layers existing, and I know that that naming scheme applies to the GI tract. So let's have a little zoom out, oopsie, and see what there is to see. Okay, so if I zoom out a little bit, let's zoom back in slightly, I can see that there is 2 layers of smooth muscle, and in this inner layer the direction of the muscle fibers seems to be longitudinal, and then up here I'm seeing groups of muscle fibers that appear to be cross-sectional. So this is, if this is longitudinal in view, that means that it's wrapping around the circle of the esophagus. So this is going to be the inner circular layer, and this is going to be the outer longitudinal layer of the muscularis externa. Now, in addition to that, there should also be some manner of outer covering, either serosa or adventitia, so it's going to be my job to figure out which. And the esophagus is basically stuck to the back of the trachea with adventitia because it's a more robust connecting tissue. So let's see what that looks like. Yeah, okay, so definitely seeing that here. So you'll notice that the smooth muscle kind of tapers off here and here. And then I've kind of got this ragged edge. So this is the edge where the esophagus has been pulled off of the trachea in order to prepare this slide. And indeed, if I go zoom in on that, I can see dense irregular connective tissue, which is what adventitia is made out of. So this is the dense irregular connective tissue of the adventitia. And I can see some adipose tissue as well, which is sometimes associated with that. So that's the sticky spot where esophagus and trachea stick to each other in the thorax. So now I'm going to navigate to the other side and see what covering exists on the front. And this looks like some looser connective tissue. So it's still lined and covered. It's not just bare smooth muscle. We have to have a place to anchor the little blood vessels that are going to supply blood to all this smooth muscle after all. It's just not as thick as that spot I showed you earlier because this area is not where the esophagus is sticking to the trachea. Okay, so let us navigate back to where we were because I want to show you some glands. I'll do this more quickly if I come over here. Okay. I like this one. Oh no, I like this one. The reason I like this one is you can see the duct associated with the gland that is going to be delivering the gland's secretion to the esophagus. So the esophagus is a muscular tube that moves food from your pharynx into your stomach via peristalsis. And food needs to be lubricated to pass smoothly from one location to the other. So in order to facilitate that, we've got glands, which are going to secrete mucus. So this is a gland. And then this is the duct associated with this gland. So this is going to produce lubricating mucus to help food move smoothly through the esophagus without sticking. Alright, so let's go have a look at what else there is to see, if anything. I thought I saw... I'm going to zoom out momentarily. Oh, wait. Nope. No, I'm not. Okay. So here we have a patch of lymphatic tissue. So all of these are lymphocytes. And let's zoom in to have a better look at these little lymphocytes. There they are. So we've got a patch of lymphocytes. Which means that this is, I'll write this in a different color so you can see, a patch of mucosa associated lymphatic tissue, or malt, as we call it. So let's take a moment to zoom out. So as you can see, even though it's pretty close to the submucosa, so the submucosa is here, this little patch is actually positioned in the middle of the muscularis mucosae, which is part of the mucosa, so it is still mucosa associated. We can also see, if we zoom back in a little bit, that it is positioned near a duct, which is right there. So this duct is probably associated with this gland. You just can't see the whole tube because it's not caught in the slice. So it makes sense for a piece of lymphatic tissue to be positioned near the lumen of the tube as a whole and near the duct, because any bacteria that you might swallow could theoretically make it into the gland by crawling through the duct. So sometimes they do that, so it's a sensible place for the patch of lymphatic tissue to be. Alright, let's see what else there is to see, if anything, but if I don't find anything that strikes my fancy, I think we're about done here. Okay. So one thing you'll notice, if you look carefully, and I realize this distinction is subtle, so up here we've got cells that are very clearly fusiform, meaning pointy at the ends, shaped like a grain of rice. So this is obviously smooth muscle. But remember, the inner circular layer is circular, and this patch of whatever this is does not match that description. Also, it's separated from the muscularis, or the muscularis externa, by this like septum of connective tissue. So that tells me that it's not the same structure. So what this is a ganglion, and it has a neighbor. And this is part of the submucosal plexus, which is a portion of the enteric nervous system positioned in the submucosa that helps to regulate the activity of the organ. So in this case, it's going to be regulating the muscular tone of the muscularis mucosae, controlling blood vessel diameter, and also directing the activity of glands. So this is nervous tissue. Submucosal plexus is nervous tissue. So let's see if we can find any other nervous tissue just before we end, because we should be able to find, hopefully, some bits of my enteric plexus, which is the nerve tissue that lives in the between the layers of the muscularis externa. And I have found some. So here's some, and here's some, and as you can see, it's sandwiched nicely between those two layers. So this is going to regulate the muscular tone and the peristaltic action of the muscularis externa. What else is there to see? There's a little bit more here, and I think that's about it. So let's just zoom back out and say goodbye to the esophagus. So that is how you recognize the esophagus as being the esophagus, and a nice tour of all of its various features. Thank you for your attention, as always, and I will see you in the next video.